Hey everyone, this is Fredrik Wormling again. Thanks for watching this yet another video about how to analyze CRISPR screen data. And now we're going to go into two tools, ImmGen and then later Human Protein Atlas. These might be a little bit more sort of special situation or a little bit more niche. Not 100% clear that they will always be relevant for your project, but as talked about before, I think it's very good when you're approaching a data set from a screen to think broadly and looking at it from different angles and to sort of try to really understand it. ImmGen is a fantastic database. It has, I think the starting point for it, or at least the way I've approached it, was to look at these gene expression data sets that are found up here. And it's typically for mouse cells, although I know now that they have more human data, where the basis has been a standardized way to sort of sort a lot of different mouse immune cell populations and then doing gene expression data. This is one caveat here then with this. So this is immune cell focused. So whether that's relevant for your product or not, you need to sort of decide yourself. But even if your study is not related to immune cells, I think it could be interesting to sort of identify if there is any immune cell phenotype or at least looking in cells in this manner. I will focus on this my gene set because this gives the possibility then to hone in on or look for gene sets. I'm gonna take the top genes here now that we found from the A549 from the DepMap data set in the previous videos, and it's a pretty short list here which will sort of affect what type of results we'll get, and keep that in mind also, and I'm doing it intentionally here, that sometimes if you put in long lists of genes, some of these tools will not work very good. So my gene set here, so there are a number of things you can select here. What's unique for the ImmGen then are these three first ones, which is either an RNA-seq, microarray version 1 or microarray version 2 data sets. And in all of these three, there have been sort of sorted a lot of different immune cells, and you can focus on some or you can just check all of them. I think it's considered that the microarray version 1 is at least the data set that contains most data. So this is the one we typically use. We're interested in granulocytes, and it's the one that has the most granulocyte data. So this is the one we're typically using. Let's check all here then. We have our list of genes, and then we can start by pressing the W plot here. And what you then see is essentially for all of these different cell populations, and they have, these names are a little bit tricky if you're not used to them. As if you start working with them, this becomes very obvious for you what it means. But the colors represent different immune cell types, and then they've been sorted in different organs and different phenotypes, etc. Here we're looking at gene expression of these genes based on different cell types. So here you can sort of see the individual genes, and what you can see is that here there's not a clear pattern that these genes would be more relevantly or more expressed in one cell type than another one. Sometimes you see that, sometimes you don't. You can download this expression data, so you get the expression for all of these in all of these ones. So essentially you get sort of these, you see value here sort of for these things. You can look at a heat map of the same type of data, and then you see all these different cells up here, and again you have to have a little bit of understanding and reading about this database to understand what the cells are, and then you see expression values that are sort of color coded here based for these ones. You can sort them in different ways. So for example, if you click the gene, or you can also click the cells, they sort of rank up. So now it's going to be from the highest expression to the lowest, for example, and you can download these as images too. And from my point of view, where my group is often interested in sort of neutrophils, I know that these are the ones I'm often honing in on, focusing on. As mentioned, you can look at only those to get more data on it. So this is, as mentioned, mouse sorted immune cells from mouse. So that's a very, in a way, a reasonably niche approach. For example, if you work with human lung cancer, for example, right? An alternative database that I wanted to go into also a little bit is the Human Protein Atlas, which is a remarkably fantastic database, and I highly encourage you to look into it. It is coming from several universities, including the university where I'm working, so therefore I'm exposed to this and I'm interested in it and know reasonable amount about it. This database does not allow for, as far as I know, to go for gene sets. So again, this will be a particular stage more in your screen setup. So this is more when you have identified a number of candidates and you want to learn more about them. Let's say we thought CDKN1A was interesting because it was strongly expressed here in this sample. CDKN1A, and then you can search for it. And the sort of starting point for this project, the Human Protein Atlas, as far as I know it, was to the very bold approach to make antibodies for all human proteins and then stain for them in healthy tissue and also cancer tissues. Since then, it's sort of expanded into a lot of different modules, but that was the starting point, and there's a lot of great data related to that. There is first here a summary. You can go into tissues then, and here you can see for most of these genes or proteins that you'll enter both RNA data and protein expression, and you can see sort of where they expressed. There's a score here, so this is protein based on these antibody stainings. RNA data, etc. further down. If you look at things like primary data here, you can see where it's expressed, and here it's actually for 2 different antibodies that they developed. So this, for example, looks very real, right? 2 different antibodies give the same data, and here you can actually go in and look at and download actual stained tissue sections from essentially all organs and all cancers, as far as I know. So fantastic data set, right? And you get information about staining here and sort of what the samples are coming from. So that's I would argue is sort of remarkable data. There is information about expression in the brain. This I think is mainly by RNA expression, as you can see here. There is single cell data. So here, if ImmGen was sort of sorting cells and then doing bulk sequence, as I understand, or microarray for that matter, here single cell RNA-seq data from a lot of different organs, focusing on a lot of different cell types, and then you can sort of get some sort of summary here also where to know where things are expressed. So very, very high resolution data, but again, you can only go 1 gene per time then. Can get ideas about subcell localization, so there's actually staining patterns with the target protein, where you can look at, and they have also oftentimes sort of 3 different cell lines, human ones. We have data from cancer, so again you can look in different cancer tissues and look for staining patterns. And again, you can get these sort of immunohistochemistry images from these things and scroll through different ones. And these have been, the histology, as far as I know, immunohistochemistry I should say, has been annotated by pathologists. So this is very highly skilled individuals looking at them, as I've been told. There's also prognostic data. So here you can see that the expression of CDKN1A is favorable prognostics in liver cancer, 2 different renal cancers. And the way that data is built up, as you can see here, is that they compare survival probability in cancers with low expression. And I think this, as I know, this is based on protein data versus high expression in this case. So low expression seems to be worse off here. And you need to look into this more to sort of fully understand the data set. For many of these proteins, they have detected levels in plasma as sort of soluble proteins, male, females, over time. They have remarkably extensive data on cell lines. Again, this is a fantastic data set, so you can look at which cell lines that the expression is highest in, both on sort of groups, but also on sort of individual cancer cell lines, right? So here you have RNA data, but also protein for some of them. Bone cancer, brain cancer. So you see there's a lot of cancer cells here, right? So you will likely be able to find your cancer cell, and if you wanna, and if you're lucky, they both have RNA and protein data on this particular protein. And in the end, there is some sort of structural information, as you can see in other databases too, and also some interaction partners based on different type of data sets. Then, so similar to what we can do with other tools. Yes. So with that, I showed how you can look for expression patterns of your genes that you found in your screen data set, both in the mouse setting with ImmGen, with the caveat that it's focusing on immune cells and that it's mouse, but you can do it with a gene set, with a list, and then secondly, using this human protein atlas to look in more or less all tissues, both on RNA and protein, but with the caveat that you can only look for one protein or gene at a time, right? And I think that could be very, very useful, but it sort of maybe comes a little bit later in a screen setup. So with that, I thank you for watching. I hope you learned something, and there are more videos in this series if you're interested in this type of material. So thank you.